Hello guys, you're welcome back to Slick Academics. In today's class, we shall be discussing meiosis. In our previous video, we discussed mitosis. Today, we shall be talking about a closely related topic, meiosis. So without further ado, let's quickly discuss some features of meiosis. Unlike mitosis, meiosis is a two successive cell division. Also, the cells here that are involved are those involved in production of gametes, especially those taking part, obviously those taking part in sexual reproduction. Also, it gives rise to 4 daughter cells, unlike in mitosis that gives rise to only 2 daughter cells. And these 4 daughter cells are haploid in nature, unlike mitosis that the 2 daughter cells are diploid in nature. Also, it gives rise to variation, meaning the daughter cells don't look exactly like their parent cell, unlike in mitosis that gives rise to clones or exact carbon copy of the parent cell. So these are the major features of meiosis. So we quickly look at the stages that are involved in meiosis. The first stage is interphase, also known as the resting stage. Obviously, here, which happens outside of meiosis, but it's also a very important part of it, because this is where there is duplication of the chromosomes or the chromatin threads. Then we also have the prophase. Then we have the metaphase, the anaphase, and the telophase. So without further ado, let's use diagrams to explain these phases as they come. So we'll look at the interphase first. Alright, so at this stage, we are going to have something of this nature. Here we still have the nuclear membrane, still very much active. And then we are going to have our chromatin threads, which are still not yet very visible. Alright, but something special happens at the interphase level. Here we have duplication of the chromatin threads. So we're going to have something like this. Now, the blue thread represents the chromosomes that we inherited from the mother, and the red represents those that we inherited from the father. There is a reason why we need to state out this, because in the subsequent stages we shall see where they come into play. So this is what happens at the interphase level, duplication of the chromosomes or the chromatin threads. Then we'll move on to the next stage, which is the prophase 1. So at the prophase 1, this nuclear membrane that we saw in the interphase will no longer be present. It will dissolve, and then the chromosomes or the chromatin threads will thicken, condense, get shorter and fatter for us to be visible. At this stage, they pair up as sister chromatids. So we are going to have something of this nature. Now they will pair up. As sister chromatids, and still they are joined at the center, known as the centromere. And also at this point we have formation of spindle. So spindle is also formed at this point. So this is basically the major thing that happens in prophase 1. But there is something quite different in the prophase 1 of meiosis than when you compare it to the prophase of mitosis. This is where we have what we call the synapses. Alright, so basically I'm going to draw this so that we see what is happening here. Now in the prophase 1, here we're going to have coming together of the homologous chromosomes. That is, these homologous chromosomes simply refer to chromosomes that control the same gene but of different variants or of different versions of the same gene, sort of. All right, so we're going to have this. Since the red represents that of the man and the blue represents that of the woman, we are going to see something of this nature. So synapses simply means coming together of this. So we're going to have something of this nature, and then when they come together in this synapse, what we call crossing over takes over. And crossing over simply means sharing of genetic material between these genes or these chromosomes, all right, which is also known as chiasmata. All right, so we have something of this nature. All these are still happening in the prophase 1. So we're going to have the both of them is already mixed, so we're going to have something of this nature. Now, do you get what we just did here? There was exchange of genetic materials, and then you can see that the genes, the homologous chromosomes no longer have just distinct features. They have mixed up and they have crossed over, sharing genetic information. All these things happen in the prophase 1. All right, so we go on now to the next phase, which is the metaphase 1. Now, in the metaphase 1, remember, there has been crossing over and they are mixed, and just like in the mitosis, they will arrange themselves in the center. All right, so we'll have something of this nature. So we're going to have something that looks like this. So these chromosomes will be aligned in the center. All right, now why are they being aligned in the center? Because in the next stage they shall be moved by the spindle to the extreme points. All right, so let's go over but first, this particular phase is known as the metaphase 1. All right, so we go over to the next phase. So here we're going to see the anaphase 1. Now in the anaphase 1, something is going to happen here. Remember, in mitosis, what we separated was actually the chromatids. But in the anaphase 1, what we are separating is actually the crossed over chromosomes. You can notice that we are not separating singly, but we are moving over the already crossed chromosomes. Now, this is one of the major differences between meiosis and mitosis. So this is the anaphase 1. Now in telophase 1, still cytokinesis is going to happen. That means the cell is going to divide. And also there will be complete division of the cell. So this is telophase 1. All right, and then we'll have independent daughter cells. All this is what we call the first meiotic division. All right, so this is the first meiotic division, because these two cells will now go on to the next division and undergo the same process. So let's quickly look at the second meiotic division. So in the second meiotic division, there is nothing like the interphase. Okay, so it goes straight to prophase 2. So I'm picking out one out of the two daughter cells, all right, because it's going to further divide into 2, and the other will also further divide into 2, giving us a total of 4. But I'll be picking out one of the daughter cells. In this case, we'll have this. Remember, they're already mixed. Okay, the reformed nuclear membrane will also dissolve, and then spindle will be formed, just like the normal prophase. So this is prophase 2. All right, so spindle will also form. We'll have spindle formation, and then the cells become visible and the rest of it. And in the prophase 2, nothing serious actually happens apart from spindle formation, and then the cells become, the chromosomes become visible. And let's quickly go to metaphase 2. In the metaphase 2, we are going to have now in the metaphase 2, we are going to have the cross chromosomes assembling themselves at the center, just like they did previously. So we'll have something like this. So they assemble themselves in the center. Of course, you know what will happen next in anaphase. Now in anaphase 2, there will be movements. All right, so let's draw out the anaphase. Now there will be movement to the extreme. All right, so the extreme, the poles. So we'll have something of this nature, and you will take notes because of our crossover materials. In this case too, the crossover chromosome, that's the chromosomes that have crossed each other and share genetic material, there shall be significant changes. You see that each of them shall be separated from their centromere points, giving rise to what we call the chromatids. But each of them shall still bear a remnant of each other on their bodies. So let's see how it goes here. Now you can see that the chromosomes that have paired, that were crossed, are now still bearing samples or remnants of the other person or the other chromosome on them. So this is the reason why in meiosis there is this variation, because there has been exchange of genetic materials between the homologous chromosomes. So this is actually happening in anaphase 2. It is now going to do what? Go over to the telophase. 2, which is the last phase. In each of the cells where cytokinesis, we also see that the cell completely divides, so we now have individual or independent daughter cells, and in each daughter cell we will have this. Now we have 2 chromosomes or 2 chromatin threads in each of the daughter cell. That is half of the number that the parent cell started out with. This is daughter cells that are haploid daughter cells. So looking at it, we can see that we actually reduce the number of chromosomes in the daughter cells through meiosis. Now, why is this important? Humans have 46 in total, and whenever there is going to be a sexual reproduction, the production of the gametes undergo meiosis. This reduces the 46 in each parent, that is in both the man and the woman, reduces the 46 to 23, so that when they come together and copulate, the resulting organism or the resulting product, which is a zygote, will be 46 in number as well. That is 23 from the parent, from the father, 23 from the mother, making it complete 46. So this is why meiosis is quite important, especially in sexual reproduction. I hope you enjoyed today's class, and I hope to see you in our next class. Thank you and stay safe.